All right, these notes will go into your AP Bio bound composition notebook. So it's going to go into the lab notebook, the bound comp book, and not the spiral notebook this time. All right, so several of these terms no doubt will look familiar to you. However, scientific method notes and scientific method terms are always important, so it's good to go ahead and review and kind of refresh our memory. So independent variable is abbreviated IV. The independent variable is the one variable or experimental condition that is purposely changed. AKA means also known as, so the independent variable is also called the manipulated variable. Independent variable is the one variable or experimental condition that is purposely changed. It is also known as the manipulated variable. The dependent variable is what is observed as a response to the independent variable. The dependent variable is what is observed as a response to the independent variable. So essentially you can think of it as what are the results of the experiment? What do we measure, count, what data are we recording? It is also known as the responding variable. So dependent variable is what is observed as a response to the independent variable. It is known as what is measured / counted / the data that's recorded in the experiment. And it's also known as the responding variable. The controlled variables are abbreviated CV. And there are other factors or variables that could have significant effects on the outcome of the experiment, so they need to be kept constant. And so that's why controlled variables are also known as constants. So controlled variables are other factors or variables that could have a significant effect on the outcome and therefore need to be kept constant. Controlled variables are also known as constants. And finally, the control, which is also called control group. So control and control group are two terms that are meaning the exact same thing. The control, or the control group, is the part of the experimental design where the independent variable is omitted. It allows you to see if the results of the experiment are due to the independent variable. In a way, it's able to answer the question, does the independent variable really have any effect whatsoever on the experiment? Not all experiments will / can have control groups. We just try to add a control or a control group whenever we can. So control or control group, that is part of the experimental design where the independent variable is omitted. It allows you to see if the results of the experiment are due to the independent variable. It can kind of help answer the question, does the independent variable really have an effect? Not all experiments will / can have control groups. All right, so I know that you've heard of a hypothesis before, and hopefully in your science career you've gotten beyond the educated guess, and we've moved up to, at the least, I hope you're writing hypotheses that are if-then statements. If I do this, then this will happen. So we're going to take it a step further. We have three different terms that are associated with hypotheses. The first is alternate hypothesis. Alternate hypothesis is written with a capital H and a subscript A. The alternate hypothesis is a hypothesis that states that there is a distinct pattern or trend in the data. It is also known as the experimental hypothesis. An example of an alternate hypothesis: for chickadees, male birds perform more songs than female birds. So it's showing that there is a distinct pattern or trend in the data. An alternate hypothesis is a hypothesis that states that there is a distinct pattern or trend in the data. It's also known as the experimental hypothesis. And an example of an alternate hypothesis is for chickadees, male birds perform more songs than female birds. So this is something that we can set up, test, record data, and analyze a conclusion for. Next is the null hypothesis. Null hypothesis, and I'll uncover this here in just a minute, but the null hypothesis is written with a capital H and a subscript O or subscript 0. It is a hypothesis that states that there is no pattern or trend in the data, or it states that there's no difference between groups, or that there's no relationship between variables, or that there is no statistically significant difference in the effect of the independent variable. So all of these things are basically saying the same thing, just in different ways. Most of the time when we look at a null hypothesis, we're either going to look at it in this fashion, that there's no pattern or trend in the data, or if we're running an analysis, there is no statistically significant difference in the effect of the independent variable. So what that means is that you can collect data. So I'm collecting coin flips. This is one of the most common ones that we use for null hypothesis and analysis. I flip a coin 100 times. I expect it to land on heads 50 times and tails 50 times. What if it lands on heads 51 and tails 49? Is that close enough to 50-50 to count? And so it pretty much is. That means that even though the numbers aren't exactly what I expected them to be, there's really no statistically significant difference in that one different coin flip. So an example of how we might write a null hypothesis: there is no statistically significant difference in the number of mates attracted by males who sing and males who do not sing. So we can go ahead and set this up and test it. We have males that have a song, songbirds that have a song and males that do not. Count the number of mates that they attract, run analysis, see if those numbers are statistically significant. So a null hypothesis will say that even though the numbers might be different, 40 and 41, when we're expecting it to be 40 for both, that one difference is just due to chance and it's not statistically significant. And finally, an explanatory hypothesis is just kind of what you would expect from that term's name. It is testable and falsifiable hypothesis that proposes explanations to account for your observed patterns or trends. So, for example, male birds sing to attract mates. We're explaining why they sing. We can set up an experiment to see if when they sing they're attracting mates. Another example, male birds sing to warn other birds of approaching predators. Again, we can set up and test that explanatory hypothesis. A few other important terms related to scientific method that you probably haven't heard of at this point. The first one would be level of treatment. The level of treatment is how the treatment or independent variable is varied or changed within the experiment. So the level of treatment is how the treatment/independent variable is varied or changed within an experiment. So for example, I want to test to see the preference of sugar for bees. What sugar solution do they prefer? My independent variable that I'm purposely going to change is a sugar solution. So I can have, let's say, 4 different levels of treatment. In an experiment to test sugar preference of bees, 4 levels of treatment, I have 4 different sugar solutions. One has 10% sugar, one has 20, one has 40, one has 50. So these would be the examples of levels of treatment. I'm testing the independent variable at different levels. A positive control is important. It helps us to see what a positive test result would look like. Focus there for me. There we go. So positive control is using a treatment to verify a known result. This allows a researcher to show that the experimental setup is capable of producing results. In a way, it shows us what a positive result would look like. So a positive control is using a treatment to verify a known result. It allows the researcher to show that the experimental setup is capable of producing results. And what I think of a positive control, I think of the fact that a positive control is going to show me what a positive result looks like. So, for example, I know Benedict's solution, Benedict's indicator, tests and shows that sugar solution is present. But I don't know what it looks like when sugar's there. So I make a sugar solution. I make a solution that I know has sugar in it, and I add that indicator to it, and I look to see what it looks like. This is an example of a positive control. In this situation, I knew I was going to get a result to show that that solution was a sugar solution. So my positive control, this is what it looks like. This is a sugar solution with Benedict's indicator added. that is helpful so that when I have unknown solutions, so I have these three solutions right here, if I have unknown solutions and I add Benedicts to each one, I know that solution number 1 has sugar present, whereas 2 and 3 do not, because my positive control showed me what a positive test result would look like. So of course, there are negative controls. A negative control is used to verify that a treatment isn't expected to produce a result. It allows the researcher to visualize when there is and when there is not an effect in the treatment. Oops, I forgot a color there. Hang on. All right, so this is what it's going to look like when it's just water with Benedict's indicator added. This gives me a result that is a negative control. So a negative control is used to verify that a treatment isn't expected to produce a result. It allows a researcher to visualize when there is and when there is not an effect of the treatment. So I know that there is no sugar present here. So what does it look like when I use my indicator solution on a substance that does not have sugar in it for sure? It's going to look like this. So that allows us to know what do negative test results look like. So when I have unknown solutions that match that color, I know there is no sugar present.